First, I'll turn on the recording. All right. So we left off last time with a poll question. We actually have a couple of poll questions. So you can go ahead and take a look at that while I'm letting the last few latecomers in. So we left off last time talking about the high throughput methods and metagenomic methods, which contrasted with kind of the earlier Sanger sequencing cloning approach. We still use both, but in general, we tend to now use more of the high throughput. And so, you know, one of the questions is, why is that, right? So which of the following would you view as benefits of high throughput genomic sequencing approaches? A, they don't require cloning before the sequencing. B, the ability to reconstruct whole metabolic pathways without or prior to culturing. C, very high coverage. Oops, sorry about that. Come on. C, the very high coverage allows the identification of mixed metagenomes, or either A and B, or all of the above. So let's go ahead and launch the poll. You guys can answer. All right, go ahead and log your answer if you haven't already. Right, that's most of you. All right, so most people put A and B. Some people, the next most popular was E. So let's take a look at that. So A is do not require cloning before sequencing. So that was definitely one of the advantages we talked about, right? And remember, when we use those methodologies, so I don't know why this is advancing. When we use those methodologies, the way they are set up performs the key aspect of cloning. And what was that? Why did we have to do cloning with the Sanger sequencing? What is the cloning achieving? Anybody remember? You're going to need to remember next week.
Is it because when you clone, you have identical replicas of an organism?
That's an advantage, but that's not really the fundamental reason why you make the clones.
Maybe
Right?
because
So that's clones in general, right? But Johan, what were you going to say?
maybe because we know the primers. Like in cloning, you actually know what you wanna extract from the DNA.
That's
And
also you know, a good thing, but
is it to identify like specific sequences, like to distinguish them between different species, I guess?
right, so it's a separation mechanism, right? So each clone should have just one gene of interest, like one copy of the 16S gene or whatever gene you're cloning. With next generation sequencing, it already does that. So the way they are set up is individual copies of, let's say, the 16S gene or chunks of the genome, right? Because remember, if we do shotgun genome sequencing, we chop it up into a bunch of little pieces, stick it onto there, and start making copies of it that way. But each individual spot will start from a single sequence, and then copies will be made off of that. So you don't have to do the cloning. Does that make sense? So that's really the fundamental advantage in terms of separation. But B, the ability to reconstruct whole metabolic pathways. We saw that in the paper we just read, right? That's what they did in the Chivian paper. So that's definitely true. Now, I know I didn't really get a chance to talk about coverage, and so C was kind of confusing, but this is also correct. So when you sequence, I did mention very briefly that you had multiple copies. So if you were assembling those contigs where they overlap, you want to have more than one copy of the overlapping regions, right? So that it's not just two sequences overlapping, it's like 200 overlapping with 150 over here. And that way you can look for discrepancies. And remember, I said that's how you identify sequencing errors as well. So that is referred to as high coverage or high copy number of each individual contig. So I apologize if I didn't use that term last time, but now we are because of this poll question. Okay, is everybody clear on what coverage means? And so we often refer to that as like, oh, 6x coverage or 100x coverage or 1000x coverage, whatever it may be. And that's just how many copies of that contig there are, or the whole genome summed across it. So which of the following would be limitations of high throughput genomic sequencing approaches, say compared to some of those earlier cloning and sequencing approaches like Sanger? Yikes. I don't know why this is doing this to me, but I guess I just have to use the, so expense, need for improved bioinformatic analyses, lack of database similarity for many genes, D, A, and B, or E, all of the above. So let's see if we can do our poll again. So which of these do you think might be limitations? All right, is that everybody? If you don't know the answer, just go ahead and make a guess and we'll get your answers logged. All right. So it looks like most everybody is going with D or E again. And those are the two that are best. So expense, next generation sequencing is quite a deal, quite more expensive. Typically, you're going to need to either do whole runs or portions of runs, and those can run into the thousands to tens of thousands of dollars for a sequencing run. The per nucleotide or per base pair of sequence cost is actually quite low, but remember, it generates huge amounts of data. So expense can be a limitation, but honestly, it's getting easier and easier and cheaper every year. So A will no longer really be an issue in the next few years, probably. But the fact that you get so much data does bring forth B. And bioinformatic analysis, and you can take a class with Dr. Berleman on this. Is anybody taking that class with him? Just curious. I know that is an elective for you guys. So bioinformatics is a course that is all about how to analyze these very complicated sequence databases. And you have to typically use coding. So I see Jenny raised her hand, so she's taking it. So you learn like our computer language, our platform, and learn how to write codes and things like that. And it's a really valuable course. So it looks like some people are going to take it next semester, and that's I would say it's a good choice of class to take, particularly because so much of what we're talking about in this field is where jobs will be in the future. So B is definitely still a limitation, but it too is getting better. And then C, I didn't really touch on this, but we mentioned how when you want to know what a sequence is, you take that sequence and you submit it to the NCBI or some database, and you ask, what is this gene, right? And we kind of glossed over the fact that sometimes when you submit those, it says, I don't know. And in fact, it can be quite common in certain novel genomes that a substantial portion, like maybe 10, even 20%, this is particularly true of viral genomes where there's so few viral genomes out there from the environment that can be an issue. So the lack of database similarity can be a problem in some cases. So Jenny, did you have a question, or were you raising your hand about the bioinformatics?
I was just raising my hand about the bioinformatics.
Okay, you might want to click your hand raise thing again so it'll go away. Or maybe it just disappears on its own, I forget. All right, so the best answer was E, all of the above in this one. All right, so let's move on and talk about quantitative molecular methods. And then we're gonna finish this portion of the lecture, and then we'll at least start the activity. And these kind of go well together, so it should be good. So if you're going to do molecular quantitation, then typically you're going to use something called a gene probe. And these are similar. You can think of them as being similar to a primer, right? Do you guys all know how primers work? Yes, no, maybe. So essentially what you have is and I'll have a picture on subsequent slides too, but you have a short stretch of the DNA, right? Or RNA, you can have RNA probes too, but let's just, for simplicity's sake, take DNA here. And so you're going to take reverse complement of the sequence, right? Because you know of the anti-parallel differentiation of the double strand of DNA. And so what you can do is design a short little stretch that is the reverse complement of your gene of interest. Let's say the 16S ribosomal RNA gene. So you can do this with RNA, you can do it with DNA. And you label that. You tag that probe somewhat. If you tag what we call a primer, it becomes a probe. So if it's just a primer that you stick into a PCR reaction, it doesn't have to be labeled. But if you are going to use it as a gene probe, then in the old days, you would use radioactive phosphate in the phosphate backbone of it. But nowadays, we don't have to do that. And we just use fluorescent dyes. So things like fluorescein, DIG or Cy3, but there's dozens and dozens of these dyes. And we saw that dyes can be used to stain whole cells, right? This is different, right? So if you're counting cells with like DAPI, that tells you it's a cell and it has DNA. What we're doing with gene probes is we're trying to come up with a specific tag so that we can label cells as being species X or family X, okay? And so we can do this at various levels of specificity, but it allows us to specifically target much more thoroughly or precisely than if you just use acridine orange or DAPI, where it's going to stain anything with DNA. And so what we do is we take alignments of sequences from our database of interest, right? And we're going to use as many of those sequences as possible to design a probe. And why do you think that might be? Why would we want to use lots of?
Is it efficiency?
What do you mean by efficiency, I guess?
Like sometimes the probes may not be successfully made, and so we prepare them in excess.
Well, we definitely want them to, so we're not making, maybe it's a little confusing. So we're not making lots of, what we're doing is we're designing our probes using sequences that we look up on the computer, and we align them, right? Remember I showed you alignments 2 times ago. And the reason that we do that is that we want to design probes that will target everybody we want to target and nobody else. So for example, let's say we want to target cyanobacterial 16S genes, okay? Or cyanobacterial nitrogenase genes. Then what we could do is download hundreds to thousands of these sequences, align them with each other, just like you were going to make a tree, but we're not making a phylogenetic tree from them. In this case, what we're doing is we're trying to find what are called conserved regions where they all have the same sequence. So every cyanobacterium has a CGA, blah, 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 blah for that. But other groups don't have that. Okay. So if you just want cyanobacteria, you would want gram positives and you would want proteobacteria and all these other groups to have a different sequence where that oligonucleotide probe targets. Does that make sense? And so what you can do is you can target specific species, you can do genus, all the way up to having just domain level probes, right? You can have a bacterial probe and an archaeal probe. And in fact, you remember when we talked about the depth profile and how archaea became more, relatively more prevalent as you went down with depth? Those studies were done with probes for bacteria versus archaea, right? So they probed cells and they said, let's count them. How many look like bacteria and how many look like archaea? So you can do it at lots of different levels on the taxonomic tree of life. So the method that was used in that case is the first one I'll explain, which is called fluorescent in situ hybridization, or FISH. And the way this works is you take cells and you fix them. And what did we do to preserve them again? How do you fix like bacterial or archaeal cells? I think somebody was trying to share, but I couldn't hear. Is there, what do we add as a fixative?
Is it formalin?
Yeah, so you can just add formalin or formaldehyde, right? So what that does is it kills the cells, but it also locks them in. The way formaldehyde and other aldehydes work is it cross-links the proteins on the membranes, and so that it keeps the cells from bursting open and falling apart. So what you have to do is you fix the cells, but then you need to make sure that the probes can get inside the cell, and so we permeabilize them. And this is typically done using ethanol. Then what you do is you add the probes and you undergo a temperature-specific hybridization. And what that is you take your probe, you add the probe into the sample. So in this case, what you can see is it's on a microscope slide. These are the cells on the slide. And then you add the probe on top of the permeabilized cells that have been prepared before and let it sit there. And you do this at a specific temperature, and that should be the annealing temperature for the probe. Okay, so that's the temperature at which the DNA should stick together. Okay. All right. I think I see some stuff in the chat. What do we got? Oh, all right. So, and then you do that hybridization. Okay. And then we're going to wash it because we want to get rid of any excess probe that was on there. You just wash it off. Okay. And then you're going to take it under a microscope, a fluorescent microscope, and you're going to shine certain wavelengths, the wavelengths you're chosen based on the probe that was used, and they emit light. Okay. And so typically when we do this, what we'll do is we will add our probe, do all of this, and then at the end, we'll also stain it with DAPI. Why do you think we would want to stain with DAPI in addition to the specific probe? Anybody have any ideas? So let's go to that example I gave of staining the cyanobacteria, and then we also add DAPI. Would that stain just the cyanobacteria, or who would that stain? Let's say this is a seawater sample. Everything with DNA. Thanks, Jose. So that's going to be all types. And so if we look at that, then we can actually see, and I'll show you, I think I have a slide on the next slide that'll show you that. So this is a schematic that shows you essentially what I just described. Right, so this is the probe. It's the reverse complement of a specific stretch, and it's tagged with one of those fluorescent dyes. This is the example of ribosomal RNA fish, which is the most common. The reason we target ribosomal RNA is that we already have all these probes and primers designed. They're really good for taxonomy, right? The other reason is that everywhere there's a ribosome inside the cell, there's going to be ribosomal RNA, because if you remember, ribosomes are part protein, part RNA. And so there's more targets for it. So usually the cells will light up more brightly for ribosomal RNA gene targeting or probing compared with just a single gene copy type thing. So you light up their ribosomal RNA, and then you're going to look at it under the microscope. Sorry, that kind of that something weird happened with that. And so then you look at it under the scope, and you're essentially looking, okay, this guy lighted up with this probe, this one with another probe, and these didn't light up at all. Or maybe they light up with DAPI. And so what you can do is you can count how many were red, how many were green, and how many were blue for DAPI. By doing this, this allows us to come up with a relative abundance, actual numerical numbers from our sample. So let's say I filtered, 100 mils of seawater, and I counted 12 of these red guys. Then I know I have 12 of that species or whatever it is per 100 mils of seawater sample, let's say. That is a true abundance, okay? And it's a specific abundance of that group of organisms. Does that make sense? And so this is what I was saying earlier. So here's a case where this is a specific targeted dye. If you do that here, it also lights up with DAPI, okay? But here, this lights up with DAPI, and we look over here, there's no corresponding cell. So what this means is that this is a more specific probe than just staining DNA. And we could count the number of orange spots, count the number of blue spots, and come up with a relative abundance of whatever it is we're targeting with this probe. Does that make sense? All right. So let me ask you a question. If you were asked to quantify the number of cells of uncultured bacterial species X, okay, we won't name it, but just some species, in a pond water sample, which would be the best approach? Would you use direct counts, fish, MPN, or D, all of the above? Sorry, I'm trying to get the poll open. So let's relaunch. What would be the best way if we're specifically interested in uncultured bacterial species X? Okay, let's go ahead and wrap it up. Okay. So most people put fish, which is what I just, so let's walk through this. All right. So direct counts, what was that again? How did we do direct counts?
You count colonies on a plate?
On a, what kind of a, on a culture plate?
Yeah.
Was that direct counts?
Maybe I'm wrong.
Okay. So on a plate, it would be a culture, like a plate count, right? So direct counts, the word direct is the clue. So why don't you try again, Cecilia? So if it's not a colony on a plate, what do you think we're counting with direct counts? Or does somebody want to help her out?
Does it just pertain to culturable bacteria as a species? Because if you take an environmental sample, there might be some which are not culturable. So direct count.
That's right. Does a direct count do uncultured ones as well?
Yes, it considers uncultured ones as well.
Yeah, so Caden puts in the chat, are we counting directly from the microscope on a, yes, on a slide. So DAPI, what I just showed you essentially, this is direct counts. Microscopic sample, it doesn't have to be culturable. We stain with DAPI, and if it has DNA, we can count it. Okay, so look back at your notes from when we talked about those, and that's what you'll see that we were doing there. Fish has lower numbers, and that's because why is the number lower on the right-hand side compared to the left?
It's specific to those bacteria that were probed.
Exactly. Okay. All right. Now MPN, why is MPN not the best answer for this question? What's the key word in the question?
Uncultured.
Probable. Oh, in the question, my bad.
Yeah. So uncultured, why is that the key word, Johan?
Because in MPN, you actually have to cultivate the species and then dilute it over a series. And for
Right.
that, they need to be culturable.
Right. So remember the MPN, we do the dilution series, and then the way we count them is how many tubes show growth in that growth medium. So if we're going after uncultured bacteria, that wouldn't work. So the best answer was B, as most of you said, but I just want to make sure everyone is on the same page with this. You guys really need to know this next week. The difference between culture counts and microscopic direct counts, fish counts versus DAPI or direct counts, okay? Because I'm going to ask you questions that require you to understand what method should be applied, and then you're going to explain how you use that. Okay. All right. Another method we can use is Qpcr. And I alluded to this earlier when we talked about PCR. You can also sometimes call it real-time PCR, because what you do is you're looking at the amplification in real time. It's not reverse transcriptase PCR. It's real-time or quantitative PCR. You can go after DNA or RNA probes this way. If you're going to do RNA, though, you need to convert it to cDNA. And how did we say we did that again? How do you convert mRNA to cDNA? What enzyme did we talk about using?
RT.
Reverse transcriptase.
So that would be RT-PCR. So reverse transcriptase, real-time PCR. That's why I hate the term real-time. And it's kind of gone into disfavor. I should probably just remove it anyway. I don't know that you'd find it in papers. So it's reverse transcriptase Qpcr. And so if you use reverse transcriptase, it converts to DNA, and then you can use PCR to amplify, like we talked about. You can do that with regular PCR, but you can also do it with quantitative PCR. What's the difference? Quantitative PCR allows us to estimate more precisely the original amount of cells that gene probe or primer set, in this case, not a probe, but it is fluorescing, so it lights up, is targeting. So we can do the DNA or RNA, as we just said. And the way you do this is you have to know roughly how much DNA is found in a cell. Like, how much DNA do you typically see in the typical E. coli cell? And you kind of extrapolate from that how many cells you had based on how much DNA you end up seeing. And so this allows you to estimate the copy number of the genes. How many genes copies were in that PCR reaction? The end number should be related to the beginning number. If you start with 1,000 copies, it's a lot easier to get to 1,000,000 copies. If you start with 1 copy, you're not going to end up with as many copies in the final analysis. Okay. So essentially, that's what we're able to do with Qpcr. If you're going to do RNA, you can also do gene expression studies, and that's allowing you to quantify how much in a sample, let's say our seawater sample we've been talking about, like for nitrogenase gene, how much nitrogenase is being made there? Well, a good way to estimate the activity is to look at the gene expression. Gene expression does not tell you that it was being used as an enzyme, but it's pretty weird to make lots of copies of the RNA if you're not going to turn those into proteins. So there are some cases where that doesn't happen, but generally speaking, we equate mRNA production with subsequent translation of the protein and the activity of that enzyme. So we can do this. So you can estimate the number of organisms, like with ribosomal RNA targeting, or you can estimate the amount of an enzyme or a functional gene that's being produced in the environment. Regardless of what you're targeting, what happens is you monitor in real time or quantitatively the S curve, right? And this is not a growth curve. Remember, this is a PCR product curve. This is true of all PCRs. With Qpcr, what you have to do is use fluorescently labeled nucleotides. Remember, we did that with the Sanger. So if you use fluorescent nucleotides that you add, then what happens is the buildup of fluorescence over time tells you how much PCR product you have. Okay. And so this allows you to monitor the increase in fluorescence. And as I said, the relative increase and the rate of increase. So how high it gets, but also how quickly it goes up. So this blue one went up faster, steeper than the sort of greenish, goldish, bronzish one. And that is an indicator that this one was in more abundance in the original sample than this one. Okay. Now you can't read very low levels, so you have to have some sort of threshold, right? You don't want to be measuring it up here because that's where it's running out of reagents and plateauing. So what you want to do is measure during the exponential, like the linear phase here of the exponential growth curve. All right, any questions about how that works? I'm not going to ask you all the nitty-gritty details of how Qpcr works, but you need to know that I can use Qpcr to estimate the abundance of gene copies in the environmental sample. Okay, whether it's a functional gene copy or a 16S gene copy. Now, like all PCR, Qpcr has caveats. So we still have the ones that we talked about before with PCR, inhibitors, primer design issues. And we also have to optimize those primers for use in Qpcr, which is a little bit trickier. With these, if you're going to estimate, okay, so I want to compare to something, then you basically need to spike your sample with control DNA, which will also amplify. If you do that, then you know how much you added. And so you can relate these numbers to the original sample and these numbers to that. And so that you know then what the numbers are as they go up and up and up for your unknown sample. Oops. All right. So we have to use those. And the other caveat is gene copies do not equate directly to numbers of cells. We have to use kind of a back of the envelope calculation that I mentioned earlier, like, oh, E. coli tends to have this many copies, so it'd be this many cells. 16S ribosomal RNA can be found in multi-copies, and it can vary from species to species. So these kinds of things can affect our results. But Qpcr is now a widely used methodology to try and come up with estimates. All right, so that is it for that. Are there any questions about FISH or Qpcr? So there's other probing methods that used to be used, but many of those have kind of been replaced by these two. So let me escape out of that. And that segues actually very nicely to our activity measurements. So what we've been talking about is how to quantify or count cells in the environment, right? And that's really helpful, right? Because if we want to know, you know, we said some of our fundamental questions in ecology is who's there, how many of them are there, right? But there's a presumption, we said, based on the how many, that things that are there in abundance are more important than things that are rare. And that's not always the case, right? So we can't always assume that. So it's important for us to also ask ourselves the question, what are they doing and how active are they in our environment? Particularly in an uncultured environmental sample, this is sort of a fundamental question that we have. So we want to explore some of the methods that we can use for this. Some of these are going to be bulk methods and some of them are going to be more targeted methods, but many of them are kind of more bulk. If you're going to target things, then you're going to use some of the abundance methods like the FISH that I talked about. And we'll talk about how the interface of some of these today. So physiological measurements, what I mean by that is activity in terms of the physiology of these cells, whether it's a bacterial cell, archaea, you know, microbial eukaryote, whatever. Our fundamental question for this lecture is, what are they doing and how much of it are they doing, right? Essentially. And so one of the things you might want to do is go to an environment and ask the question, how active is this process in that environment? But one of the issues that we have is that you often want to know what our baseline would be in an undisturbed environment. So many environments have been impacted by humans. Really, you can't go anywhere on the earth and there isn't some sort of human impact. But there's certainly places that are more impacted than others, right? So if you sampled in the middle of downtown LA, that is not an undisturbed environment. Versus if you sampled in the middle of the central coast of California, that is a much less disturbed environment to sample in. And so that's kind of the things we can think about. But disturbance is also really tricky. So for example, nutrient disturbances can happen invisibly, right? There may be eutrophication that has occurred upstream of something and we didn't even know it happened. So for example, nutrients might be greatly impacted remotely, or air pollution might be, or global change at a global level might be impacting these environments too. So let's put it this way, it's getting harder and harder to find undisturbed environments, but we can try. And then we just have to acknowledge that disturbance is a big part of it too. Now, I make a lot of points over the course of this semester about human disturbances. And of course, we are major drivers of that. But it's not always us, right? So there are natural disturbances that occur. Weather existed before climate change, right? So weather impacts matter. And we can't always identify, oh, this was a human driven event, or would it have happened naturally without? Forest fires, wait, we know they're getting hotter, bigger, and it's likely because of our impacts. But forest fires have been going on in the state of California since there was fire in California, right? So that's really kind of the challenge of understanding things, but we just have to try to assess what's going on. And if you want to look at degradation due to anthropogenic impacts or recovery after anthropogenic damage, that can be a little bit challenging, but we may want to have a reference site which is more undisturbed. So for example, if we want to study coastal Long Beach or just Southern California Bight area, which is this, area between Point Conception and down to, South Orange County, then, maybe we go take a sample out in the ocean, far away from land, and that could be our undisturbed control. Okay. So if we're measuring activity, we have a number of methods to do it. And perhaps the most commonly used is radio tracer studies. So what you can do is take a sample and you can add a known quantity of a radioactive substance like carbon-14, right? And that allows it, what it does is it labels your sample and allows you to track the movement of the sample or the transformation of the sample. So a classic example is heterotrophic potential. What do you think I mean by heterotrophic potential? If I took a soil sample and brought it back to the lab and did this, I'm measuring heterotrophic potential. What do you think I'm really asking? What's my question?
What kind of like how many species, I guess, in your sample would be capable of using organic carbon? Is that what you're asking?
Yeah, I don't know that you'd identify the species that are doing it, but you'd be asking the question of how much, like if you're doing a soil, what would we figure our major carbon cycle would be in a soil sample, assuming it's oxygenated? What's our fundamental thing? Like if we're using glucose, let's say we want to know how much glucose is broken down inside a soil. What metabolic pathway would be breaking down the glucose?
Respiration?
Exactly. So heterotrophic potential is essentially equated with respiration potential. It could be aerobic respiration or anaerobic, depending on conditions. And so what we can do is we can C14 label glucose, take a pipette and spike it into our soil sample. And if we're going to look at respiration, what do you think we want to look at on the other end of the equation? Literally the stoichiometric equation. We add glucose, that could be in a water solution, but what are we going to measure? What state of matter?
Gas.
Carbon dioxide?
Exactly. CO2 gas. Good job, guys. So what you could do is spike a sample into a bottle and then trap the gases that are liberated from that bottle and measure the amount of C14 CO2 that was in there. So if it started off as C14 glucose and converts to C14 CO2, boom, I added this much glucose and I got this much CO2 out. And you can do a mass balance equation just like you guys learned in general chemistry, right? So these are the kinds of things that we do. We can also look at just the transformation, not the uptake necessarily. And so that's what that is, right? Converting glucose to CO2, not necessarily using the glucose for cell synthesis. But you can also look at uptake. right? So did that carbon get integrated into the cells? And for example, photosynthesis is often measured via the uptake of that. Now, the way we measure radioactivity is not with a Geiger counter, right? Geiger counters are not sensitive enough. So typically what we need to do is take our sample and we put it in something called a scintillation counter. And what that does is a scintillation fluid lights up when you put radioisotopes in there. So if you have cells or chemicals that are radioactive and you drop them into this special fluid, you can then put it in a machine and it will count the number of photons of light that get released. Scintillating, you may have heard that term before, that's light release. And so that's what it is. You can also do something called microautoradiography, and I'll show you a picture of that in a minute or two. So anytime you do these uptake experiments, we have some assumptions. One is that you're doing linear uptake over the time course of incubation. And so typically what you have to do for this is you need to do a time course. And so maybe you spike your sample with the C14 labeled glucose and you measure CO2 release every 10 minutes for 2 hours. Okay. What we want to do is calculate a rate of uptake or rate of utilization, right? And so if we're looking at a rate, that's a slope, right? Something over time. We want our slope to be linear. If you let it get out of linearity, then what happens is it levels off, just like we've talked about with our growth curves. And when you're leveling off, it's not going to be an accurate measure. So linear uptake is our assumption. And then we also need to know what our specific activity is that you've added. What does that mean? How much radioisotope did I add? Okay. This matters in terms of some of these mass calculations. I'm not going to go into the details, but just be aware that specific activity is something to care about. All right. So what I talked about briefly, mostly thus far, was just transformation, but cell synthesis is another thing we care about. How much cell growth is there in an environment? Well, in a spectrophotometer with a pure culture, we can measure cell growth by turbidity, right? Easily. But with an environmental sample, you can't do that. I can't take a lake sample, drop it in a spec, and tell you, oh, this is how much cell growth there is going on in that lake. But what I can do is I can measure cell synthesis using, in this case, tritium, another radioisotope, right? So the tritium labeled thymidine. So thymidine will be incorporated, right, as one of the bases, right, as T's, right, thymines, into cells, into their DNA backbones. And so what you can do is measure sort of a little bit indirectly how much cell growth there is based on how much DNA synthesis there is. And there should be a linear relationship between DNA synthesis and cell growth, because of course you have to replicate your DNA every time you make a new cell. So this method of using tritiated methylthymidine allows you to do that. And it's fairly commonly used in aquatic samples. Now what we do is we spike our water sample with the thymidine, incubate it for a period of time, and then we extract it afterwards. The assumptions here are somewhat similar to what we saw before, but also additional ones. We're assuming only bacteria are uptaking this if that's what we're interested in, but there might be other organisms in your water sample that are also taking it up that could skew that. But if you like filter your samples with appropriate filters, you can make it so it should be bacteria. There's an extraction step, and the assumption there is that about 80%, it's 80% efficient in its extraction, okay? The other expectation is that the level of ambient thymidine is insignificant. And that's a good assumption in most cases, because if there was thymidine available, it would be slurped up and used in DNA synthesis by any organism. The other assumption is that T's make up 1/4 of the 4 bases. So what you do is you look at thymidine incorporation and multiply everything by 4 to understand the total DNA synthesis. And then we also have to estimate how much DNA there is per cell. Because ultimately, just like we saw with Qpcr, right? We don't really care about how much DNA synthesis is going on. What we care about is what that means in terms of how many cells are being produced, right? And so there's this assumption that this is 10 to the minus 15th or 10 to the minus 16th, right? So this is the amount of DNA per cell in grams. So it's really tiny, right? You're not going to measure that on a balance, but it's been estimated. All right. Another approach to use that does not use radioactive methods is the BRDU method. So instead of using thymidine, they use this bromodioxyuridine, right? Uridine like uracil. So remember, T's and U's are very similar. And so what you do is instead of incubating with tritiated thymidine, you put this bromodioxyuridine in. The cells aren't smart enough to realize this is not thymidine, and they just slurp this up and use it. And what you can do is use an antibody probe to the BRDU, and the amount of probing from the antibody should be the amount of BRDU inside the cells, and then you can do similar kind of calculations. So this is a non-radioactive approach for cell synthesis. All right, I know this is kind of a lot, so are there questions about that? So the take home from this is you're using labeling of nucleic acids as a way of estimating the number of cells that are being synthesized via DNA replication and then, you know, going through into fission, right? Binary fission. Okay. You can also look at RNA, and so you can tritiate adenine to look at RNA. You can look at protein synthesis by using tritiated leucine or amino acids like leucine. Okay. All right. So that's like cell synthesis growth type things, but you can also look at inorganic transformations. And that's sort of what I talked about in terms of the heterotrophic potential earlier. The opposite of that can also be true in terms of C14 can be used. Before we were looking at CO2 production. In this case, we're going to label the CO2, or usually we label bicarbonate because you can have a liquid solution of it. And if you want to look at, say, photosynthesis rates, you're going to add C14 labeled CO2 and try to follow it as it gets converted into sugars, okay? Or cellular incorporation. So you can take a water suspension, and I actually did a lot of this in my doctoral work because I was looking at cyanobacteria, and I wanted to ask questions like, how are their photosynthetic rates impacted by exposure to ultraviolet radiation? So I would take bags, fill them with cells and water, and I would spike it with the C14, close it up so it didn't escape. And then over time, I would then measure how much of that C14 got converted to cell biomass. I would filter the cells, and then I would put them in the scintillation counter. Okay. One thing you have to worry about here, which we don't have to worry too much about, is the alkalinity of the water is related to how much CO2 is dissolved in it, and so you have to account for that. Another method that I've used that's very commonly used is to look at anaerobic respiration. So sulfate reduction, as we've learned, we've talked about, right? So under anaerobic conditions, these organisms breathe sulfate, converting it to hydrogen sulfide, which is shown here. So in this case, what you do is you purchase sulfate solution with the radioisotope of sulfur, which is 35, S35, and you add it in to your sample, and then you measure the sulfide that's produced. And just like with the heterotrophic potential, remember, sulfide is going to be liberated as a gas. The way to determine how that happens is it's complicated. And so there's apparatus like this, where you've got these distillation things where you're bubbling. These experiments are actually kind of dangerous. So you've got heavy metals, you've got radioactivity, and then you boil it all. So it's at high temperatures for these distillations. So it's kind of, but you can successfully measure sulfate reduction that way. Now, one of the assumptions of these methods has to do with that specific activity that we mentioned earlier. What you need to make sure is that you're not changing the amount of the substrate, the reactants in the sample appreciably. So if I take a huge amount of radioisotope sulfate and add it to a sample that has very low sulfate concentrations in it to begin with, I can skew the stoichiometry. I don't want to do that. What I want is my radio tracer to be a very small fraction of the total amount of the reactants. But then what we can do is measure the transformation of the S35 or the C14 over time and use that to extrapolate then for the total amount of sulfate or CO2 that might be converted in the environment. So we don't want to overall change the concentration of the reactants. We add just a little bit. And the good thing about radioactivity is we have such sensitive ability to measure this in a scintillation counter that you don't need to add very much. And in fact, radioisotopes are really expensive, so you don't want to buy very much. Because of course, what I hope you all are aware with radioisotopes is they all decay, right? That means they disappear. Now, the good news about C14 is that the half-life is measured in thousands of years, so you have a long time to use it. But for example, for S35, the half-life is about 90 days. So if I buy S35 for my lab, 90 days from now, half of it is gone, even if we've never run an experiment. So those are things to consider as well in these experiments. All right, I mentioned that another way of looking at how much radioactive activity has happened is through something called microautoradiography. And so what you do is you take a sample and you spike it with a radioisotope, C14, for example, like we said before. But instead of using a scintillation counter to measure the amount of radioactivity being given off, what you do is you add a special solution. And it's kind of like the silver emulsion solution that we used to use with old school photography. So what you do is you bind the radio tracer to this emulsion and you get a precipitate. And can you see these little dots here? So these are little precipitates. And what this showed, this is a study, this is actually a cyanobacterial cell. And what this is an example of, a study was asking the question, where does the radio tracer go? Does it go into the cyanobacterial cell, or does it go into bacteria that are living on top of the cyanobacterium? And what you can see here is that each of these little dots is a little heterotrophic bacterium. And so in this case, they identified that it was the heterotrophs that were incorporating the radioisotope, not the autotrophs. So that's just an example of that. So you look at this under a microscope and you just count dots. Now, you can also combine microautoradiography with fish. So, a few minutes ago, I told you about fish. So fish allowed us to do what? What was the purpose of using fluorescent in situ hybridizations? What did that allow us to do? Have we forgotten from an hour ago? Somebody remember, please.
Allow us to label the DNA of our target organism.
Okay, and we were going to use that labeled target organism. How? Like, what were we going to do with those?
In that example, we were measuring against like DAPI or direct counts. Just numbers, finding out how many are
So
there.
we're trying to enumerate, right?
Yeah.
All right, so fish allows us to count stuff. Microautoradiography allows us to know who's using a substrate. And so what you could do is design a probe, fish probes. It wasn't done in this study, but what you can do is you can probe the bacteria with fish probes, and you could ask the question, this organism is using the C14. What is it? And if it lights up with the microautoradiography like this, and it lights up with like a specific gene probe for 16S genes of a specific genotype, then you could ask questions like, okay, well, we know there's heterotrophs here, but which heterotrophs are using it? Is that a gamma proteobacterium that is the one that's utilizing it? So you can combine those in the MAR fish. So most of what I've talked about thus far are radioactive methods, except for the BRDU one that I measured. But there are non-radioactive methods as well. So you can measure the production of gases, like we've talked about CO2 release and sulfide. You can do that using glass microelectrodes. And this is actually a study from Yellowstone Hot Springs with a couple of good friends of mine that were using this microelectrode, which could be lowered down into this cyanobacterial mat that we were studying. And in this case, they were measuring oxygen. And what you can see is that as it went down, the oxygen level went way up, and that would have been due to what in this mat? Why would oxygen go up near the surface of a cyanobacterial mat? Anybody know?
Can you repeat the question, please?
Yeah, why do you think
Would it be phototrophs?
Yeah, so they're oxygenic phototrophs, right? So cyanobacteria produce O2 during metabolism, during the day at least. So they were measuring this huge spike of oxygen, but then what you notice is it goes way down as you go deeper. And what that was due to is heterotrophic oxygen consumption, respiration, as you went deeper into the mat. So that's just an example of that. You can also use colorimetric assays and titration assays to measure things like oxygen and sulfur. This is a so-called Winkler bottle. The other thing that you can do is study stable isotopes. So radioisotopes are the decaying isotopes, right? But what you should remember is that for most of the simple elements, you actually have multiple isotopes. You may have a radioisotope of it, but you also have two stable isotopes. So let's take the example of carbon. C14, as we just saw, is the radioisotope, right? And so it has a half-life of a few thousand years and it decays, right? And disappears and converts to another element. The normal element that you guys learned about in chemistry for carbon, what's the atomic number of carbon normally? Anybody remember?
12?
12, right? So C12 is what we normally think of as carbon. And most carbon on our planet, in your body, in the rocks in the ground, all around us in the trees, is C12. But it turns out that there's a little bit of C13 as well. Okay. So that stable isotope is heavier, right? It's got that extra neutron. And so it is by mass heavier, but it's also distinguishable chemically. And so we can actually trace things using stable isotopes in kind of an analogous way to what we did with the radioisotopes. It is a lot harder though, because radioisotopes are really easy to get a nice clean signal from. The downside, of course, is you have to use radioactivity, which can be very dangerous if not handled properly. So the plus of stable isotopes is you don't have to worry about radioactivity. You're not in danger at all from this. But to measure it, you usually have to do something like gas chromatography, mass spectrometry, okay, to analyze that. But fortunately, in scientific research centers, even like at Cal State Long Beach, there's GCMSs on campus that can be used for this. So what you would do is instead of spiking with C14, you could spike a sample with C13 and then measure its transformation or its incorporation in much the same way that we did. Usually for this, you need larger amounts of isotope because it's harder to detect and longer incubation times. Now, one of the really cool approaches that was developed in the late 90s, early 2000s, I guess early 2000s, is called stable isotope probing. And what you do here is you add either C13 or nitrogen 15 to cells while they're doing their thing, incubating, growing, and it labels their DNA or their RNA as they are producing new cells. You can then extract, and you don't have to do this with cultures, right? You can take a sample from the environment, incubate it with the stable isotope, and then extract the DNA or RNA from that sample. You can then purify that extracted sample using a cesium chloride gradient. Remember I said that's a good way to clean DNA and get it ultra pure. And so in this case, what happens is the organisms that took up the C13 into their DNA will have heavier DNA than the organisms that didn't. And if you use an ultracentrifuge, you can actually separate on a cesium chloride gradient the nucleic acids of the unlabeled cells from the labeled cells, right? So the cells are dead, they're lysed, we've done a DNA extraction, but in here should be the DNA and ribosomal RNA of the utilizers, and in here, it's not there. Now we're going to do this in a mixed tube, and essentially you can see you separate them. So what we can then do is go in and take a needle, suck out this fraction, suck out that fraction, and we can sequence them. So if we PCR amplify, let's say the 16S gene from the black band and the red band, and do next generation sequencing with it, or even just Sanger cloning, whatever, you can then identify the organisms in this band and compare it to the organisms in that band. And that allows you to ask a simple question. Who uses isotope X, right? Who's growing and using that, okay? So this is just a very brief description of a study that a former master's student of mine, Lindsay Darjani, did when she was in my lab. Did anybody have Lindsay for general bio? I know she teaches a lot of our general bio classes still on campus. So her master's thesis was centered on asking a question. Who uses lignocellulose, which is very complex plant carbon in a salt marsh sediment? So what she did is she spent a lot of money buying C13 labeled lignocellulose. It was like thousands of dollars for like this little vial of reagent. So it was super precious. She was sweating that one. And then she inoculated her samples and let them sit there for about a month. And then what she wanted to do is ask the question, okay, where did that carbon go? And so she did the SIP experiment, just like I showed here, right? Where she separated things out. And she sequenced the different fractions. And this is what she found. So in looking at the specific groups, there were some groups that were found here and here, like this pseudosporellum was at about the same percentage in both fractions. We don't really care about them. But let's look at this group. This group of sulfate reducing called desulfosarsina was very heavily represented. It was 6% of the users, okay? It's not even here in the light ones. So they must have been in the sample to begin with, but what we saw was that this is an example of a group that seemed to have been overrepresented in the utilizers of the C13 labeled lignocellulose, okay? And so we can then draw hypotheses from these where we ask questions like, what's going on with these guys? Or similar, this group of spirochetes, which are those corkscrew labeled cells or shaped cells that are very commonly found in marine sediments. They weren't real abundant over here, but they were very abundant over here. So this is a way that we can get at utilization and activity without knowing who's there to begin with. Does that make sense? All right. All right, so in terms of other activity methods, we don't always label DNA. We don't always use radioisotopes. Sometimes we can rely on fairly indirect methods. So nitrogen fixation is a very important process. We've touched on it multiple times this semester already. And we talked about how it's important in the transformation of nitrogen on our planet, et cetera. So N2 is a weird chemical, right? So N2 is defined by its chemical bonding structure. And if you look here, you'll see that it's triple bonded, right? When you took chemistry, you learned that triple bonds were kind of weird, right? Very rare, super strong. And this is partly why nitrogenase activity and nitrogen fixation is really energetically expensive, because you got to break a triple bond every time. So it turns out that the radioisotopes for nitrogen are very short-lived and very dangerous. So we don't typically do nitrogen fixation studies with a radioisotopic of N. Instead, what we do is an indirect test. So if you take N2 gas and add it to a sample and then ask the question, where does NH3 come from? The problem is N2 gas is everywhere, right? So we can't possibly add nitrogen gas to a sample and change the concentration of it in a meaningful way. But it turns out that acetylene is very rare. And acetylene is kind of the carbon equivalent of N2 in the sense that it has a triple bond. And nitrogenase enzyme is dumb. Nitrogenase enzyme has evolved to cleave triple bonds. And so what you can do is take a sample and add acetylene and watch the transformation of acetylene into ethylene. And that tells you how much nitrogenase enzyme activity you have, because the nitrogenase enzyme thinks it's converting N2 to ammonia, right? And so we can actually measure this very simply and cheaply in a gas chromatograph. And these have very distinct peaks. Acetylene and ethylene are easy to see on a GC. And this gets around the issue of the fact that N2 gas is everywhere. Does that make sense? So this is another non-radioactive method, and it's an indirect method, but it works very well. All right, so I think that I will stop there. We're almost done, but I do have a poll question and another slide to go. So we'll just finish off with those next time. So on Monday, what I'd like you to do is bring questions. I want you to start studying for the midterm this weekend. Okay. You want to be reviewing primarily your PowerPoint notes and everything we've talked about to this point. I will say that you guys have a lot of work to do based on the slow responses I got today when I asked questions about stuff we covered last week or a couple of weeks ago. I'm a little concerned, but you guys can get there. I believe in you. But what you need to do this weekend is you need to study. And what I would encourage you to do is for all of this stuff we've been talking about recently, all these methods, right? We've got our counting methods, both quantitative and generalized ones. We've got the molecular methods. We've got the activity methods. What you want to be thinking about is if I gave you a sample and asked you, how do I measure X, right? So for example, if I asked you, how would you count the number of species X that I showed you in that poll question earlier? That is a perfect example of a question I could ask you on the midterm, right? And if you put MPN as your answer, you're going to get some points because you at least put a counting method, but you're not going to get full credit because the question said uncultured, right? So it's those kinds of scenarios that I'm going to lay out for you. And on Monday, we're going to have a review. I'll give you some examples as well, but I really encourage you to study over the weekend, and then you can bring your questions and I'll give you that review session. So we'll finish up the last couple slides first, and then the rest of the 75-minute period, you can ask questions, or we can kind of go over these scenarios before our midterm on Wednesday. The other thing is we can talk about logistics of the midterm, right? So it's a little bit unusual because it's online, right? So it's going to be, the midterm will be posted on Beachboard. You'll have essentially the class period time to do it, okay? And then you have to upload it to the Dropbox, right? So it's a take home in that sense, but it's a limited time take home. So it is open notes, okay? But you're going to need to be able to rapidly look through your notes, choose those answers that answer the questions, right? So I'm not going to be asking a lot of really simple, easy questions. I'm going to be asking, more complicated questions because you'll have your notes to look at. So you don't have to memorize a lot of the nitpicky details, but what you need to do is familiarize yourself with the material so that you can reference it to answer the questions. Does that make sense? So I know it's tricky. It's not normally how we do things, right? If it was in person, I'd just be handing you an exam and it'd be closed book. But given the challenges of COVID, and so it's open book, but you can't talk to each other. And I will caution you, last year I had people that talk to each other. And guess what? It's real easy for me to tell if you talk to each other, okay? Because we're going to use the comparison software when you upload this, okay? So it's real easy for me to know if you guys are sharing answers with each other. I want you to do your own work. And that's part of why I made the midterm worth less, right? The midterm is only worth 75 points. So please don't cheat and risk your grade, right, for 75 points. I want you to study and bring questions on Monday, and we'll go over this, and you'll get the opportunity to then, demonstrate to me that you're kind of understanding these methodologies, as well as the different habitats that we talked about earlier in the semester, what organisms are there, what their adaptations are, all the way back to the beginning of the semester. So I know it's a lot of material, but you don't have to memorize all the minutia, right? Because you've got your notes, but you have a short amount of time to do it. So you need to know where things are and where to look things up as you formulate your answers. Okay, so you can't not study. If you try and take this test without studying, you're going to be spending the entire time flipping desperately through pages trying to find what I want you to find, and you're not going to get it done. Are there any questions about the midterm or what we're doing Monday? Oh, is it 50 on the syllabus? It was 75, but let's see.
Yeah, I just looked it up on the syllabus and it said 50.
Oh, okay, it's even less, right? So now it's only 50 points. So thank you for correcting me on that, Hamza. So it's 50 points. So it's basically an opportunity for you guys to demonstrate that you're paying attention, you're following along, and you're understanding. Like, I also want to know how much you guys are getting out of this. Okay. But it's not going to kill your grade if you get a C on the midterm. Okay. You have a lot of other points. So do your best and study hard this week, and then we'll get through the midterm next week. All right. So Sandra, you asked, is it take home or during class hours? It is a take home during class hours. So it's a little bit unusual. I will post it and set it on Beachboard that you can download it at the time of our class. And then you have until the end of class to re-upload it. Okay.
Thank you, Dr. Dillon.
Mm-hmm.
Professor,
Yes.
could you go over that poll question with me really quick?
Sure. Which one?
The DAPI one, the uncultured X one.
Uh-huh. So
I thought since it was uncultured, it would be more beneficial to use the more non-specific one. So the direct cell count.
the direct
Can you
cell
explain to me why that's not the case?
Yeah. So the key there, that's a good question. I wish you had raised your hand in class because I could explain to everyone. So what I was asking for was species X, right? Now I didn't say what that was, but let's say it's a species of Bacillus. Okay. So if you want to know how many Bacillus subtilis cells are in that soil sample that you're studying, why would a DAPI count not tell you that?
Because it's accounting for everything else in the sample,
Right, because DAPI is going to stain everything, right?
not only Bacillus.
Bacillus and
Yeah.
everything else that has DNA. So what you would have to do
Yeah.
is go to your database and look up all the Bacillus subtilis gene sequences you could find, and like 16S genes, right? As it turns out, you can just go online and Google Bacillus subtilis 16S gene probe and find it. But you'd get a probe for that specific organism. Now, Bacillus subtilis is cultured, so it's a little bit of a misnomer. Let's say uncultured Bacillus X, right?
Yeah.
Because when it's an uncultured organism, oftentimes all we have is a gene sequence from the environment and no cultures. And so that's the case where you can go to the environment, PCR amplify the 16S genes, and then if there's an organism you're interested in, like let's say the desulfosarsina I just talked about with my master's students project, right? That desulfosarsina is not cultured. It was from dirt. So let's take a gene probe now for desulfosarsina and go to the environment and ask the question, how abundant is it? Right? If we did that, we could ask the question, how abundant is desulfurcina species X? If we did DAPI, it would be a few of those cells, but it would be all the other cells in there as well. So that's why a direct count of just DAPI
Okay.
would not be the correct answer there. It wouldn't be the best answer. It is, however, better than the MPN
Yeah.
answer in the sense that the MPN answer would only be cultured organisms. And so that's really, that would be very problematic.